So good morning. So thank the organizer for invitation to share our work. So this, this is the third Syngap research conference I've been to. So according to Mike, really I've been to all of them. So lucky me. Yeah, I really enjoyed the past two, and today we're really off to a very good momentum. So I try to really keep up this good work. So today I will talk about this molecular mechanisms, our new work on this, how the Syngap gene is regulated. So just to give a context what we're going to talk about, so think of your genome as a dictionary, right? So there are about 22,000 protein coding genes, think of as words. Actually, this number is quite similar to the English words in the dictionary. So this every cell get to pick, which gene, which words to choose to compose their article. And the message of life that really determines this functioning of the cell has a lot to do with the amount of the words that was picked. That's the gene regulation. So in the case of Syngap, so this is really regulated by this in the neurons. So here there's a work done by my teammates Shuchi and Madeline. So we really did a developmental trajectory characterization, how this Syngap gene is regulated in the neurons. So as you can see in those cultured mouse neurons, in the first week or so, really there's a very little amount of Syngap within those neurons, in this shaded area. And really in the second and third week, you begin to see much more Syngap and eventually have this synaptic localization, right? So this quantified numbers are right there. So this developmental trajectory, almost like clockwork, is very much tightly regulated, suggests that there's an intricate mechanism that really controls how much Syngap gets made. And we study this not only as a biological or scientific curiosity, understand developmental biology, but we think about this, these two alleles of this gene, think of as two assembly lines churning out RNA and protein, right? So in a normal neuron, there's two assembly lines providing Syngap. But in the case of this mutation, where really one allele gets mutated, you can think of one assembly line get decommissioned. So then you don't have enough RNA and protein products, right? So, but one thing to keep in mind, in this haploinsufficient case, there's still one assembly line still intact. So if we can somehow, we can persuade that genetic allele to work overtime, right, to produce more of this RNA and protein products. We can use, especially if we can have a drug that can do so. So then that will solve this your Syngap deficiency from the molecular base, from its very root cause, right? So then all those downstream, like this very important functions, synapse or non-synaptic, will fall in line, right? So this, so I would need with. So the color is a bit off, but the message is still there. So this, so I will start with good news. So we already have a drug. We already have a drug that able to stimulate the expression of Syngap genes. So and we'll share a lot of our recent work really to where do we go from there. So first is we try to understand how does it work, right? Within the neuronal context, this drug can increase Syngap expression. How does it work? What's the molecular mechanism? And also importantly, as any drug for the CNS, for the brain, kind of really enter the brain, right? What's the brain penetrance? What's the safety profile? Right? So is this particular molecule safe? Is this particular pathway that's a molecular target? Is it safe to target? And then that we share with some of our ongoing work where do we go from there, right? So let's start off. So we have so here shows a structure of a molecule found that can stimulate Syngap. So as to how we found it, let's just say it's kind of like a bycatch when we were fishing for drugs that stimulate my favorite gene called KCC2. So we published this a couple of years back. So the way this drug works is binds to like a transmembrane protein called F33. That's a receptor tyrosine kinase on the membrane. So and it blocks its activity. So what we found is in the both in the mouse and the human neurons, is able to stimulate the messenger RNA expression substantially, and also can stimulate the protein expression. So Syngap is shown as a third name there. So really in a dose-dependent manner, significant dose-dependent manner. So really what I'm showing here is that this somehow this small molecule drug that binds with a protein on the membrane, block activity, can change the Syngap gene expression in the nucleus. So what's the mechanistic connection, right? So think of it as your gene. So think of it as your genome, how it's regulated as a control panel that's have 22,000 keys, right? So that's all the genes. So this what's the connection? There's behind this board, there's a lot of wiring. So how do we find this particular gene that able to light up the Syngap gene expression? So this is similar to what my son does to my laptop until he found the caps lock key, right? So, but how does it work this in a biological lab setting? So Shushi and Martin developed this basic genetic screening profile. Let me walk you through. So the concept is pretty simple. So we have this drug, right? So we have a drug, we put it on the neurons, and we sequence, this RNA like from this neuron. So we understand what gene overall, what's the picture of genes regulates. So we use this nominate candidates. So candidate genes will satisfy two criteria. So first is called transcription factor. Those are genes that control the expression of the genes. And second is respond to a drug treatment. In this case, we particularly pick the genes, the transcription factor genes that gets downregulated by the drug treatment, right? Then we try to recreate that downregulation of this transcription factor through a technology called CRISPR, which we can design certain codes that allow us to knock down this candidate transcription factors one by one in the neurons, and then we clone those CRISPR reagents, transfecting neuron, and basically assess Syngap expression. So through the screening, so it's still ongoing effort, but what I'm just sharing what are the recent finding. We found that a couple of transcription factors, once you knock down their expression, the Syngap level actually go up in the neurons. So what this means, so including a couple called ETS1, STAT5, and RUNX1. So what it means is that those transcription factors in the neurons, their endogenous level, their endogenous function is to suppress the Syngap expression. So once you're able to either use a drug to downregulate or use a genetic approach to knock it down, then the suppression gets lifted and Syngap expression go back up, right? So we really then this really give us some mechanistic understanding how this drug works. But then the important question follows is how good is this molecule as a drug to treat brain disease, right? Because before this particular compound was developed as a leukemia drug, was actually went through a phase 2 clinical trial, have a good safety profile in the leukemia patients, but does it enter the brain at all? So we did this so-called PKPD, pharmacokinetics, pharmacodynamics work. So basically, the idea is we supply this drug to the mouse, either through the IP injection or through oral routes, and we measure the amount of drug in the blood sample and in the brain sample at different time points. So what you can see from these curves here is this drug is already available. Basically means you give it to the mouth, it still can be absorbed, right, and go to the bloodstream. And also you can have a half-life of just a couple hours, which is very similar to what the clinical trial, the earlier work showing in the blood cancer context. And importantly, we measure the brain concentration of the drug and show the drug able to penetrate the brain, enter the brain at the concentration that's therapeutically beneficial, right, as we determine in vitro. And then we look at this, we collect mouse brain at different time points and see where they can engage the targets, where they can induce the gene expression changes. So here I've shared the results of my favorite gene, the KCA2. So this showing that this drug is actually, keep in mind the drug gets washed away from the system with half-life of about 3 hours. But then the effect of stimulating the KCA2 expression can last up to 3 days. So that's long after the drug gets washed off. So it really shows this, it's kind of this gene regulation type of therapeutics, able to kickstart a mechanism that's able to produce more protein. And also this protein itself have a long half-life. So we are still working on the Syngap. What I can share with you is this drug indeed can work on Syngap in vivo in the mouse, but time course is somewhat different. It seems like it's actually can last much longer. Its onset is slower, but can last much longer in terms of Syngap. So there's more results to come. So then we really thinking like for this KCC2, my favorite gene here. So Syngap is a close second now, but for the KCC2 is an important regulator of the excitability of the neurons. So the prediction is you have more KCC2, the neurons, the mouse should be more resistant to seizure. So we set up experiment with those mice with the drug, the concentration of the drug. This particular dose we give is much lower. It's like actually 20% of what tested in leukemia clinical trial setting. So it's actually a very low dose. I'm going to give it just once, right? And then we test basically the EEG. We set up EEG recording with the help of Dr. Jamie McGuire from Tufts University. So what we found is that with just one single dose of the drug as a monotherapy is really able to suppress the kinase acid induced seizure in a very substantial way. So not only the load of seizure, the total amount of spikes is reduced, but also 2/3 of mice were protected from this so-called status epilepticus. That's like a very strong seizure, right? That really caused the rewiring of the brain and also caused like very long-lasting detrimental effects. So we're able to suppress with just one single dose injection of this drug, right? So this. So then we want to really look into this, we understand the mechanism and we know this drug can enter the brain. So really want to understand how good is this F83 as a therapeutic target for the brain. This was used to develop for blood cancer, right? It's a lot of work in the blood world, but in the brain, really nobody understands where it's expressed, what's the function, etc. So we did a lot of work to really go down this rabbit hole. So this, what is known is that this is a receptor tyrosine kinase on the membrane. And we are the first to show that in the neuron, it's really expressed in a neuron-specific manner. Sorry, in the brain, neuron-specific manner. So there are many cell types in the brain, but this F83 is showing this red, you see this red almost like donuts. That's really co-localized with neurons, but not with associated with microglia. And this is really evident in this beautiful staining from Yu Qi, my colleague, in the cerebellum tissue. So especially in the GABAergic neuron express high level of this of the F83. So receptors there. And also what this shows is that the drug enters the brain, it actually have a cell type specificity, meaning that the receptor it blocks only present in the neuronal cell type, right? So that really gives can restrict its side effect. And also it went on to really look at this. So this is, like not mouse genetics we do, but basically the message is we develop a mouse allowing us to knock out this gene. So instead of using a drug to block a fraction of this protein, we actually eradicate this gene from the genome. Just say, okay, if you block 100% of this receptor all the time, so what's the side effect? What's the problem? So what we found is overall this mouse is actually there's no overt problem. So the body weight looks similar, the behavior movement looks similar, the grip strength, all the things we look at actually looks very similar. Looks very normal in the mouse. But when you challenge knockout mouse with a PTZ, that's a seizure induction type of reagents. So these mouse really are protected against seizure. So they look pretty normal, but somehow this knockout this gene can protect them against the developing seizure. So as you can see from this behavior called the recent scale, time course, basically you scale you score this behavior abnormality, seizure related abnormality. So really having this gene knockout, really able to block this very severe seizure from this mice. So I will quickly wrap up and summarize what I have showed you. So so we mapped out a genetic program, right? So molecular program that connects this receptor tyrosine kinase F83 on the membrane, which is druggable, to the expression of Syngap, so through that transcription mechanism. So this not only really fills a gap into understanding how this gene is regulated, but also those transcription factors represent potential therapeutic targets. And also we can leverage this information really to understand on the genome where those transcription factor binds. Right, so are there some genetic mutations that's in the non-coding region, but regulates the expression of the Syngap? So there's really open up a rich field of investigation. And from the therapeutic development angle, so I hope I really convinced you that F83, this kinase really people study a lot in the blood cancer, but really can be repurposed for brain disease, especially for the epilepsy disease. Drug development is a safe target, it's expressed on the neurons, and also we have good ways to target this receptor in the brain. So one slide, where do we go from there? So within my lab, so I have two very talented senior researchers in my lab, really following up this work, Katarina and Aridesh. They really to use the human, use human haplogroup sufficient neurons to study. Okay, are we able to restore the Syngap expression in those neurons, and are we able to rescue the synaptic abnormalities, which is my lab's really expertise in that. And we are collaborating with people. So we're collaborating with Graciela Di Cristo, I believe is also a SRF founded researcher in Montreal. So she has good characterization of a Syngap mutant mice. So we test our drug in their mice setting. And also we're working with the Shop Academy in the Axonis therapeutics who have a good KCA2 targeting drugs. So we test those in the Syngap context. right, good. So I thank all my NAPS people along the way. You meet all of them. I really thank SRF for the support and thank you for the attention. Yeah. Good.